Hi, this is a version of a talk given by me, Tim Churches, at the 2026 Triennial Platypus Conference held at Taronga Zoo. This talk was given on the 11th of June 2026. So it's a talk about thermal cameras for detection of platypus in smaller waterways, and it's describing work done by Oberon Citizen Science Network, and the co-authors Alan Sheen and Louisa Jorm. So, in terms of context, Oberon Citizen Science Network was established a few years ago, located in the New South Wales Central Tablelands, about 125 kilometres west of Sydney, at altitudes of around 1100 to 1350 metres above sea level. So it's actually higher than the top of the Blue Mountains, with a subalpine climate. There are several rivers in the area which are absolutely famous for platypus populations, particularly the Fish, Duckmalloy and Cowmung rivers. And looking in New South Wales Bionet Atlas and the Atlas of Living Australia data, it was clear that there were many historical observational records and surveys in the lower reaches of these and other rivers in the region, but there were relatively few records more recently. And in particular, there are very few or no records for upper reaches or headwaters of any of these local rivers. And it appears no systematic surveys of those headwaters or upper reaches at any time. So this is just a chart showing platypus and ricali observations in Oberon LGA as recorded in Bionet Atlas. You can see that they're relatively sparse, despite the large populations potentially in some of the rivers I just mentioned. And there was a flurry of activity in the early 2000s, but much less more recently. This chart doesn't show our work yet. We're yet to contribute observations we've made of platypus to Bionet Atlas. So based on this, we formulated a typical citizen science research question, and that is, what is the extent of platypus occupation in the upper reaches and headwaters of these rivers around the Oberon local government area? And there was actually there is some urgency in investigating this question due to a proposal to build what is by far the largest terrestrial wind farm in Australia in state forest pine plantation areas south of Oberon Township, where in fact many of these local rivers have their sources where they rise. The proposal is for more than 250 8-megawatt turbines, each about 300 metres high. That's about the height of Centrepoint Tower in Sydney, with a 2 gigawatt total nameplate capacity. And although such facilities can be built, there is a lot of potential for adverse impacts on local hydrology, and in particular on remnant native vegetation tracks, which are in preserved riparian tracks, which crisscross the plantation pine forests. Forestry New South Wales and the developers have characterised the plantation pine forests as sterile monocultures, but in fact they are crisscrossed by remnant native vegetation, mostly around the waterways, which are in fact the headwaters of all those rivers I've just mentioned. So this is a map of Oberon, and the green dots you can see are proposed wind turbine locations. Yes, there's a lot of them in the brown areas, which are pine plantation areas, state forests. But if we overlay that with the gazetted protected riparian areas, you can see that there's quite a lot through the pine forest. But in fact, even these gazetted areas are very incomplete, we found. They seem to have been defined by some automated process about 10 years ago, and only named waterways are shown, and even then, not even all the named waterways have been gazetted. So in fact, this vastly underrepresents riparian areas in these state forests. So in trying to undertake such a survey, we quickly determined that relying on direct observation by volunteers was not really going to be scalable. These target regions, I've just shown you on the map, are really quite remote. They're very large. It's about 50 or 60,000 hectares in the pine forests shown on those maps. And access can be quite tricky, and in particular the weather in winter can be quite adverse with snowstorms and blizzards even. Capture and tagging methodologies for platypus are not really available to citizen scientists due to lack of expertise and the need for ethics and ethics oversight and animal welfare oversight. eDNA would be very useful, but it's still quite expensive and a little bit outside the realms of citizen science organisation without external funding. But we asked ourselves, could we actually build some do-it-yourself imaging technologies which might be both affordable and also more effort-effective in detecting platypus in these waterways. So our first attempt involved the use of commercial time-lapse cameras, and this was based on positive results reported in 2024 by Simon Roberts and Melody Serena in Australian Mammalogy Journal. So we acquired 2 time-lapse cameras as recommended by them in that paper, Brino TLC 300s. And we tested them on a 1 kilometre stretch on the upper reaches of the Duckmalloy River in Chatham Valley, which is about 15 minutes drive south of Oberon. And this particular stretch of river had been observed by us for several years, with a very occasional fleeting sightings of a platypus at dusk, just 1 or 2 times over several years. But we deployed the time-lapse cameras in August 2024, and on the very first occasion, recording at dusk and dawn at 1 frame per second, and we got immediate positive results. And in fact, every deployment captured between 5 and 10 clear visualizations of platypus at dusk, which probably represented several individuals in that 1 kilometre stretch of the river. So that was encouraging. And this is an example from our camera. This is actually the upper Duckmalloy River, and you can see here is a typical V wave of a platypus swimming downstream in this relatively calm section. The river's about 10 metres wide, but here you can see the platypus actually approaching the camera, and it's clearly visualised there in the time lapse. But what we found is that time-lapse cameras were completely ineffective once night fell. Their low light and no light performance was particularly poor or non-existent. And we also found that we made no observations at dawn, suggesting that the platypus had returned to their burrows by then. So we thought maybe we could just use artificial lighting to overcome that difficulty, but that was ruled out due to being overly disruptive to the platypus and other animals. Unfortunately, most commercial time-lapse cameras include a fixed internal infrared filter, which removes infrared light and makes the video look, or the recorded images look natural during daylight, but it means that you can't use artificial infrared illumination at night. Artificial infrared illumination is desirable because it's largely invisible or completely invisible to mammals, but it can't be used with cameras that have fixed internal infrared filters. And yes, we did actually consider artificial UV lighting to make the platypus's pelts fluoresce, as was reported widely in the popular media several years ago. But a few calculations revealed that the power requirements were quite prohibitive for field use, and also there was quite a lot of potential for harm to platypus and other animals if bombarding them with high intensity ultraviolet light all night. So our next generation attempt was some do-it-yourself cameras, this time using special sensors, and artificial near-infrared lighting at 850 nanometer wavelength. So our Mark I camera used a Raspberry Pi single-board computer, which is a very small computer about the size of a deck of playing cards, and a special Sony StarViz camera sensor, which has no infrared filter, and a large aperture wide-angle lens. And all of that was powered by a 12-volt lithium camping battery with 6 watts of near-infrared light, which is just a commercial light designed for use with security cameras, all housed in a waterproof enclosure. And this is the inside of that Mark I camera. You can see it's quite simple. The battery's also inside the enclosure, not shown as the infrared light. And the Raspberry Pi was configured to provide its own Wi-Fi hotspot. So for field use, and an Apple iPad was used to connect to this hotspot in the field to set up the camera, so the camera could be aimed and focused correctly. And so on, all of that run using custom code, which we wrote ourselves. Not fantastically user-friendly, involves typing Linux terminal commands on the iPad, but nonetheless it works well enough. And we started shooting at 3 frames per second, which was much higher than the commercial cameras offer of 1 frame per second, the time-lapse cameras. But after that we quickly progressed to shooting full motion video at 25 frames per second. So this is our first generation camera mounted in a tree on that same stretch of the Duckmulloy River. And this is me holding an iPad, and you can see that's the camera view shown on the iPad with the camera just behind it. That allows the camera to be positioned correctly. And here we've got some examples. First of all, time-lapse, and then full motion recorded with this Mark I camera. So you can see here a platypus coming upstream. This is, I think, at 5 frames per second. You can see the platypus is probably aware of the camera. It possibly can see a dull red glow from the infrared lights, even though the actual light itself is invisible. So it's a bit curious and hesitant, but it does come upstream, and there's another platypus, same spot, heading downstream, clearly time-lapse. And here's the same camera, but this is on Yellowbelly Creek, which runs into the Duckmulloy River, close to where we were filming previously. But this is where Yellowbelly Creek is actually running through the state pine forest. So this is along a remnant tract of riparian area, remnant native vegetation, even though it's clearly overgrown with a little bit of blackberry. And here's a pool, just and in front of the pool is a culvert that goes under a forest road. And you can see we get very nice vision of the platypus. Again, it seems a bit curious, dives. And if you watch carefully, it goes up through and the culvert under the road. And you probably didn't notice, but in the foreground, this waterway is also full of galaxids, native freshwater fish. We haven't identified the species yet, but it's probably a large species complex. After that, we tried a next generation Mark II cameras, basically the same design, but using better, even more sensitive Sony sensors, higher quality lenses, including a short telephoto lens to allow the camera to be positioned a bit further away from the waterway, because we were worried that the camera itself was scaring the platypus, and larger external batteries. And the results of these cameras were good, but not a huge advance on the Mark I camera. So here's an example of using the telephoto lens. So the camera is actually set back possibly 10 meters from this viewpoint, but the infrared lights are positioned a little bit closer. And you can see it nicely captures the platypus almost totally submerged underwater. Near infrared light penetrates water to some degree, to about 10 or 15 centimeters depth. But we found, again, some issues with these cameras. As I mentioned, the slight visible red light emitted by the near-infrared illumination, or possibly the camera enclosures themselves, were being spotted by platypus when they surfaced. And their reactions range from cautious curiosity to sometimes an outright startle response, and diving again rapidly. The whole setup itself is quite heavy and cumbersome to deploy. And there are high power requirements of the near-infrared lights, so 6 watts each, and using 2 lights plus the camera using another 6 watts means that we only really had limited duration of about 1 night from a 30 amp battery, which is approximately as much as one can easily carry. And the video file sizes were absolutely enormous, and reviewing the videos was very, very tedious, and it was actually very easy to miss a platypus as it flashed by, particularly if heading downstream. We did try some naive computer vision assistance code, which I'll show you in a second. But we also last year undertook a collaborative project with Masters of IT students at UNSW Sydney to train AI models to automatically detect platypus in these videos and distinguish them from ricali and ducks and other animals. But that project was only partially successful, mainly because we didn't really have enough training material at the time, but also using video like this really requires quite specialized AI techniques that can make use of the full motion video, not just individual images. So here's, and again, a Mark II camera, but this time we're running some fairly simple computer vision detection code in the camera, and so it actually marks the platypus as it goes past, as you'll see. Unfortunately, it's not an AI model, so it marks basically anything that goes past, and it would also mark up a duck or a ricali in the same way. Nonetheless, that's a potential advance because we don't have to watch hours of videos just to find that little snippet as the platypus or some other animal of interest goes past. So, having done those experiments and found some limitations with the near-infrared cameras, we wondered whether we could actually use long-wavelength infrared sensors instead. So these are a class of sensor that uses a vanadium oxide microbolometer to sense black body or thermal radiation in wavelengths between about 8,000 and 12,000 nanometers. So the near-infrared is 850 nanometers, just outside the visual range of mammals, but instead thermal cameras only sense thermal radiation at much longer wavelengths. So these sensors operate at room temperature. They don't need cryogenic cooling or anything exotic. They don't consume very much power, less than 1 watt, and they work in absolute darkness. They need no artificial illumination of any kind, not even infrared. And in fact, they're completely insensitive to visible light. So these sensors used to be very expensive, but the cost has dropped dramatically, along with the specialized germanium lenses that need to be used with over the last 5 or 6 years. Thermal radiation is absorbed almost completely by glass, hence the need for specialized germanium oxide lenses. But also thermal radiation is completely absorbed by water. So even 1 or 2 centimeters of water is almost completely opaque to this black body thermal radiation, which we suspected might have been a problem since platypus tend to like and live in water, but we thought it was worth a try. So we first of all tested a relatively cheap low resolution thermal sensor, around $300, with a 9mm lens, obtained via AliExpress from a company called Shenzhen Thermal in Shenzhen in China, interfaced to the same Raspberry Pi computer that one of our existing near-infrared cameras. In fact, we just retrofitted it to the camera, so we're actually able to record both thermal and near-infrared video imaging of exactly the same subject area at the same time. But to our surprise and delight, on the very first night of deployment, on a calm stretch of the upper Duck Malloy, we were able to clearly and quite unambiguously identify a platypus swimming on the surface near the camera. And this is the very first capture on a thermal camera of a platypus. You can see it's clearly a platypus. It's not a ricali. It's got platypus anatomical features. The movement is typical of a platypus, in particular that diving action. You might not be convinced by that, but I'll show you some higher resolution thermal videos shortly, which will convince you. So there are a few issues with the Mark I thermal camera, and that is that the low resolution was not very good, and the field of view with a 9mm lens was way too small to be useful in smaller waterways. So we stepped up to a higher resolution thermal sensor, also with the same 9mm lens, but that provided a much wider field of view with the larger sensor. And as of September last year, when we first obtained one of the high resolution sensors, they were about $850 Australian from China. But in fact, exactly the same lens and sensor setup is now $600, and the cost still seems to be falling. So these are now very affordable bits of equipment. So we put that together in a camera in the same way, interface to a Raspberry Pi and storage and so on. And this provided much better thermal imaging video, as we'll show you in the following examples. So This is the first iteration of that. This is one of our existing near-infrared cameras in the larger box, but we've attached the thermal camera, and you can see the germanium lens peeking out in the smaller box at the foot of the tripod, so we can record both near-infrared and thermal. This is the later generation of a thermal camera. This is a pure thermal camera. It doesn't have near-infrared recording as well. You can see at the bottom the germanium lens and the Raspberry Pi, a solar interface, so we can actually charge this from solar panels, and storage on the left, solid state drive storage. Still very much do it yourself, but relatively easy to construct and highly reliable. And this is that same camera. I mentioned it doesn't support near-infrared imaging, but the smaller box also contains an ultrasonic echoacoustic sensor, a microphone, which is able to record ultrasonic bat calls as well as bird calls, frog calls, and even potentially platypus vocalization in the wild, if we ever find a platypus that vocalizes, which apparently they do sometimes. And you can see the thermal lens is actually protected by another glass element. This is a germanium glass element just to keep the water out. So this setup is completely waterproof and could actually fall in the river without any damage. So this is one of our first deployments. So this is at the junction of the Duckmalloy River and Yellowbelly Creek. This is showing the deployment. So there's the near-infrared camera and the thermal camera, and because we're using infrared as well.
This is the section of Yellowbelly Creek shown in the thermal imaging.
Wide, so
about 1 metre wide.
But where we're focusing on this little section of the Yellowbelly Creek where it's about 1 metre wide and running downstream into the Duckmalloy. And here's some of the thermal. So here's a platypus. You can see it's a platypus, two widely spaced eyes, quite distinct from a ricali, heading downstream from the Yellowbelly into the Duckmalloy. And a few hours later, here's another one. No platypus were observed going upstream, so we know that's definitely two platypus coming downstream from the quite small Yellowbelly Creek into the Duckmalloy. So here's some video on the same section or similar section of the Duckmalloy, upper Duckmalloy River, and this, just to orient you, the river's here about 10 metres wide. The reflections you see are actual thermal reflections of the surrounding trees in the river. So here we've got video. And this is actually a bat, a microbat, I'm not sure what species, flying above the river. You can see its hothead and the reflection. But now if you watch the left bank about a quarter away from the top of the frame, you'll see there's a platypus surfaces, and you can see very nicely the not just the V wave it makes on the surface of the water, but a thermal plume that it leaves behind it in the water. So this is not the platypus heating up the water, it's rather the platypus mixing warmer lower layers of the water with the cooler upper layer of water. Here's another platypus coming downstream, and again a lovely thermal plume. There you can see some ducks, and this is actually a ricali, just shown to distinguish it from the platypus. We know it's a ricali because the swimming motion's different, and the head looks different, and the head is held right out of the water rather than sitting flat on the water. We also know it's a ricali because it's clearly chasing the ducks, and we've discovered through this thermal video imaging that ricali are completely badass when it comes to ducks, and harass and harangue them and attack them all night long. So here's the ricali coming towards the camera, and you can see it's quite different to the platypus. And here's a platypus coming across the river, so just to distinguish it from the ricali, again you can see two eyes there. Characteristic swimming motion and a characteristic diving motion as well. And another platypus coming upstream in the middle of the channel there. So, very effective at the higher resolution, and this is not a small waterway, it's about 10 metres wide at this point, but I've also shown it in the Yellowbelly Creek where a waterway about 1 metre wide. Here's again on a larger water body. This is actually a dam on Duns Creek. This is located deep within Gurnang State Forest, so this is an area of remnant native vegetation. completely surrounded by many kilometres of plantation pine forest. So it's quite surprising to find platypus here, but there they are, they, as you'll see in a second. And this dam is actually formed by a forestry road which crossed Dunn's Creek and formed this dam. Dunn's Creek actually runs into the Tuglow River, again quite famous for platypus, and the Tuglow River runs into the Calmung River and then downstream into Canangra Boyd National Park. So you can see a flock of ducks in the background, but in the foreground you can see there's a platypus on the surface, and it's just dived. And same spot, slightly later in the evening, and a platypus surfaces from under the lilies, just in the foreground. Very clearly a platypus. Now here's using the dual near-infrared and thermal camera just to capture video, and this is the upper Tuglow River. So this is about a kilometre downstream from that dam that we were just looking at. The channel here is maybe 1 metre or 1.5 metres wide. So this is really at the upper reaches of the Tuglow River. This is also deep within the state forest, surrounded by plantation pine forest. And you can see there's a platypus with the characteristic hot eye sockets. Probably just looking at the dull glow of the near-infrared lights. But this is a thermal scene, so this does not rely on the near-infrared illumination. And in fact, if the illumination was not there, it would look exactly the same. In fact, the thermal sensor is insensitive to the near-infrared illumination, just as they are insensitive to visible light. But here is exactly the same platypus at exactly the same time, recorded with the near-infrared camera, doing exactly the same thing, but you can see that in fact the vision of the platypus is not nearly so clear, and that could be mistaken for a fish, for instance. But we know it's a platypus because that's exactly the same animal at exactly the same time as on the thermal. So, a few limitations of the thermal cameras. First of all, they're probably not useful if the platypus is likely to pass the camera while completely submerged. More than a centimetre or so, because water is definitely opaque to thermal radiation. But also the thermal contrast between the platypus and the water may be a lot less in warmer climates. These were shot around Oberon over summer, but even in summer the water there is quite chilly, and the waterways remain cool. In winter the water becomes very cold, we've measured down to about 2 degrees, so just above freezing. Where we expect there to be very, very good thermal contrast between platypus or ricali and the water, but in other areas that may not be the case. So we've, I think, proven, at least to our satisfaction, that thermal imaging is a viable methodology for detecting platypus in these waterways, both larger areas and small waterways, provided the observation spot is carefully chosen and the platypus is not going to be swimming entirely underwater. But there's lots of opportunity for future development of this technology. Longer focal length lenses are available for these sensors, and in fact we have tried 18 and 35 millimeter lenses. And these may be useful when you know exactly where to point and focus the camera. So for instance, surveillance of a known or suspected burrows, where we can catch the platypus going in and out of the burrow. But we've also considered and started to try automatic tracking of platypus on the surface of larger expanses of water using computer vision algorithms, similar to the one I showed you earlier, running in the camera on the feed from the thermal sensors to control a motorized pan tilt mount, so that the thermal camera can actually track the platypus on the surface of the water using a longer wavelength lens, longer focal length lens. It may also be feasible to run computer vision algorithms or even simple AI recognition models, again on the Raspberry Pi computers in each camera, to detect potential platypus and or ricali sightings, and that would reduce the data storage requirement a lot. which would allow longer deployments and also reduce the human review burden once the cameras are retrieved. But I've already mentioned the thermal cameras don't use much power, and they don't need any artificial lighting, which also uses a lot of power. So we could already run these cameras automatically to record for 3 or 4 nights in a row before our current batteries run out, and we're currently testing solar charging of those batteries using a PV Pi, which is a nice little add-on for the Raspberry Pi developed by Auto Ecology in Melbourne. Each camera can also be configured to record visible light if we wanted to, so we could record during the day. That's not really a requirement for where we are. Platypus just don't come out during the day, but I know that's the case that they do elsewhere. And I've already mentioned simultaneous echoacoustic recording. It makes sense to record at least bats in that environment as well. And it's also easy to add other sensors to these do-it-yourself cameras, so air temperature, air pressure, humidity, rainfall sensors, water temperature, water turbidity, or even underwater still or video cameras triggered by the platypus approaching as detected on the thermal camera from above. And then finally, intriguing prospect is that because these thermal cameras allow undisturbed behavioural observation and they're quite cheap, it may be possible with some modest funding to, if you like, instrument a whole section of waterway. So setting up thermal cameras placed perhaps every 100 or 200 metres along a waterway over a 1 or 2 kilometre stretch, and then record continuously over weeks or possibly even months. And the timestamps on the videos recorded can be synchronized via GPS satellite clock signals to within at least a millisecond of each other. So it would be possible to very accurately track movements of platypus up and down, which may allow for accurate estimation, or at least statistical estimation of the number of platypus in that particular waterway. All right, thank you. That's the end of the presentation.